Okay, so what I covered there in the first part was about this basic structure stuff, the divisions within the nervous system. Now what we're going to be looking at is what nervous tissue is. So nervous tissue is one of the big four, epithelial, connective, muscle, nervous tissue. We usually think of nervous tissue as being specialized for communication in the sense of electrical signals traveling through our body. It's not the only tissue that is responsible for communication, but that's sort of its primary function in the sense, no pun intended, of sensory input and motor output. Okay. Now, nervous tissue is made up of two types of cells. There are neurons, which we're going to take a little bit of time to talk about now, and then, come on, glial cells. It's just those two. Sorry, yeah.
I have a quick question.
I have a quick answer.
Okay, well, good. I just have a question. My friend was that kid that was hit and had a lot of that hit and run.
Okay, I'm not familiar with the story.
Well, he was hit by a drunk driver, so he severed his spinal cord.
Uh-huh.
So they're saying right now that he's paralyzed from the chest down.
Uh-huh.
But they said he also has slight movement in his right hand.
That's because the nerves that go to your arm are these right here, which are coming out of the base of your neck. So if he had the spinal cord injury somewhere here.
I thought it was the neck. I thought she said it's severed somewhere up here.
So these nerves are coming out of all of this part of the neck. If he had the spinal cord injury here, then there will be some input that goes to parts of the arm that might be intact, and then other parts that he might not have full use of his arms or full strength in his muscles, and then nothing else.
Which could come back or no?
No.
So saying that he's paralyzed from the chest down is
Yeah. Sort of the best known example of that sort of spinal cord injury is Christopher Reeve. And he was an advocate for spinal cord injury research, and he was very involved in pushing the envelope in trying to get his neurologists and other doctors to help find the best way to get some recovery of function. He never got full recovery of function, but there were some good signs of things later on. Some of that research is probably making its way out into general practice these days or something like that. But some of it's still very cutting edge. One thing that's very big in making those sorts of things work is stem cell research, which has its own sort of political issues to deal with. Also, I'm going to be talking about these glial cells, and they play a role in nervous tissue repair, and that's sort of the reason why central versus peripheral do that. And there's some possible therapies from that I'll mention. There was a study that was published sometime the past year about actually getting spinal cord function back. I can't remember if it was in a human or if it was in an animal model, but whatever it was, a very significant leap forward to trying to get that stuff back. But how close that is to actually being used in humans, I have no idea. But I'll talk a little bit about some of that stuff. So, at its simplest, nervous tissue is just made up of these two types of cells, neurons and glia. Neurons are sort of the main ones that we associate with this electrical signaling. We're going to talk about what their structure is in a bit. Glia are the supporting cells, and there are different types of glia. There are different types of neurons, but there are different types of glia that play different roles in supporting the nervous tissue. And so we'll talk about that too. Now, as I'm talking about these things, I'm actually going to sort of integrate in with that this laboratory material by looking at some micrographs that I've captured from the slides that we have. I'll show them up on the board. I'm pretty sure that all of these slides that I want to show you, is this too dark?
No.
You can handle that?
Yeah.
Okay. One, it'll be easier to see the screen, and two, your brain will go, wow, it's dark in here, it must be nice and cool, and it'll make you feel a little bit better. The sweltering heat we have here. But anyways, so I'm pretty sure that the slides that I want to show you are in Blackboard. You'll be able to get to them. I'll check that in a second. But first off, to talk about what the neuron is. Whoops. A neuron basically has three major parts to it. There's the cell body, and then two different types of things sticking off the cell body. Collectively, those two things that stick off the cell body are called processes, or processes sometimes. The word process, meaning a thing sticking off of another thing, in its most general sense, is actually something that's been used already in anatomy. When you were looking at bones, there were some bones that had markings that were called processes. It sort of depends on how deep you got into the information in the skeleton when I was doing it. But for instance, in the skull, there's this very thin little piece of bone sticking out right here. Okay, the skull's upside down. That's where a particular muscle attaches, and that's called a process. It's a very thin little spike of bone sticking off of that. That's called a process too. Okay, so when we're talking about a process, its most general definition is something sticking off of something else. Okay, bone processes, cell processes, whatever it might be. Now, the processes that we have sticking out of a neuron cell body are either dendrites, which are all of these sort of very branched things that we see here, or the axon, which is this one thing sticking off here. For the sake of drawing a picture for a textbook like this, it's often misleading. While the axon in this picture looks like it's pretty big, pretty thick, they're actually very, very thin fibers. Okay? Also, axons tend to travel quite a distance. Now, when I say travel, I don't mean that they physically move in your body all the time. When that neuron originally was born in your nervous system, the axon grew from the site of the cell body out to its target. And that fiber is basically set in place your whole life. So when I say travel, I'm really kind of implying the developmental growth of that thing. But they can go quite a long distance. There are neuron cell bodies in your brain that go down through your entire spinal cord, even to the bottom of the spinal cord, which is right here, around the small of your back, the lumbar region. It's one fiber, the whole distance there. It grew there originally when that cell was born, back in during development. So they can be much longer than this picture suggests. And they're also going to be much thinner relative to the size of the cell body itself. But there's only ever one axon coming off of the neuron. Now the axon can branch. So just like there's one wire coming out of the light switch over there in the box, it branches into all of the different light fixtures up on the ceiling. Okay. Same way, axons have a unique source, their particular cell body, but they can branch and connect to a bunch of different targets, okay, which is not really represented in this picture, but that's the case. Generally speaking, dendrites are what we consider the receiving end of the neuron. And in this picture, kind of faded out a little bit, is the end of another axon that's connecting to the dendrites here. So it's receiving information from another source, and then that information in the form of electrical activity is going to travel across the cell to the axon, where it'll travel down the length of the axon to its target. And where it connects to its target, it forms what's called a synapse. And when we were talking about the muscles last week, I introduced the neuromuscular junction. That is a type of synapse. Okay. In this picture, we're looking at neurons connecting to other neurons, which happens quite a lot in the nervous system. But there are neurons that connect to skeletal muscles at the neuromuscular junction. There are neurons that connect to cardiac muscle, or to smooth muscle, or to glandular tissue. So whatever the target is, the connection it makes there is essentially a synapse. There are some variations in what synapses are like, but we're not going to get into that too much this semester because that's mostly relevant in A&P 2. So for the sake of this semester, we're going to just talk about the synapse, and it's going to be very similar to what we learned in the neuromuscular junction, which is just a specific type of synapse. So that's the neuron, the cell body, or soma, which is just the Greek word for body, which contains the nucleus, and then everything sticking off of it, which is mostly dendrites, and then the one axon. Also in this picture, this sort of blue-green translucent looking thing, or things, are insulation around the nerve fiber. Okay, just like the wires that go in the circuit to the lights above us, there's insulation around this. Okay? There is a difference. The insulation around an axon is not a continuous, complete surrounding cover. There are gaps between little bits of insulation. So one segment of insulation, like we see here, or here, or here, or here, here, is called the myelin, or the myelin sheath. Okay. This cell body off to the side, that's the same color as that insulation, this is a glial cell body, one of the types of glia that we're going to learn about a little later on. And it's responsible for extending its own processes out that wrap around the axon and provide the insulation. There are 5 different segments of myelin represented in this picture, but it's drawn so that only these 2 are actually part of this this glial cell. The other 3 are probably part of some other cell. Okay, doesn't really matter at this point, but there will be little segments. There are gaps in between those segments, which are called nodes of Ranvier. So Ranvier is just some dead French guy's name, which you pronounce it with that kind of, French, I'm too lazy to pronounce the end of the word kind of thing. So that's Ranvier. That's their full name. I'll sometimes just call them nodes for the sake of brevity. But there is a little gap between the myelin sheaths where the axon cell membrane is exposed. And that'll be very important to how the electrical signal travels or is conducted along the axon, which we'll be talking about next week. Now, I make this point in the book because I think it's very important that these pictures are very misleading. I already told you that the axon really is thinner and longer than this picture suggests. It's not drawn to scale here. Also not drawn to scale is the size of the myelin sheaths that we see here. Okay. For the scale that we're looking at, one myelin sheath should go way off the side of the screen here. Okay. And I'm going to show you a micrograph of myelin a little later on so you can get a real sense for how long it is. But for the sake of the drawing and the book, they make them look very small, very short. Okay. And I consciously didn't ask the art editors to change that for this picture because trying to make it look more realistic would actually make the picture a little harder to fit on the page and get all the information into it that you want. So I'm willing to accept this shortcoming in the drawing, but I do make a point in talking about it in the book, and I'm going to make a point talking about it tonight, that this is not really what that looks like. Okay. But that's the basic structure of a neuron. Okay. The glia, which provides the myelin sheath, is not part of the neuron. It's another cell acting as support. And in this particular case, it's the support it provides is the insulation around the fiber. Now, these pictures look very simplistic on purpose. The idea here is there are different types of neurons, and we classify them at their simplest into these three groups. And so I just had these very cartoonish drawings made to depict these. The cell that we were just looking at is basically what this cartoon here is suggesting, okay? But there are a couple of other variations. Now, these are all called something polar neurons. A neuron has polarity to it. It has a receiving end and it has a sending end. So the unipolar, bipolar, or multipolar nature of a neuron really is referring to how that polarity relates to the actual cell body. So this thing called a unipolar neuron has only one process sticking off of the cell body itself. And that one process is the axon. Now, unipolar neurons are essentially this shape, but very different in their size and connections. One end of the axon, which might be this end, and for the sake of the cartoon, I didn't make a real big point about it, are the dendrites. So this is the receiving side of the cell. And then the axon continues on past where it comes out of the cell body to where it's going to make synapses with its target. Okay. The length of the axon here, sticking off this unipolar neuron, is very, very out of scale. Unipolar neurons, generally speaking, are found in ganglia along the length of our spinal cord, and the dendrites are found out in the periphery. A lot of them are in the skin. And then the other end of the axon, synapses in the spinal cord or brainstem. So if you have a unipolar neuron that has dendrites in the tip of your big toe, its axon can go all the way back the length of your foot, turn up the ankle, go up the knee, up the leg, and then the cell body will be located in a ganglion right here at the base of the spinal cord, again, in the lumbar region, the small of your back. And then the axon will continue on into the spinal cord. Okay, so it might be coming in here, and the cell body is at the base of the spinal cord, but the axon continues up the spinal cord and synapses in the brain. Okay. So for me, that axon might be 2 meters long, roughly. Okay. I'm not 2 meters tall, but it has to go the length of my foot and then all the way up to my brainstem, which is right about here. Okay. So roughly 2 meters long. That's probably the longest axon that you'd have in your body, going from the tip of your big toe all the way up to your brainstem. Okay. And that would be a unipolar neuron. Okay. Now, calling this a unipolar neuron is actually incorrect. This is a pseudo-unipolar neuron, which means that there's only one thing coming off of the cell body, but that one thing branches into two things, okay, or branches into two directions, so that there's dendrites at one end and synapses at the other. Okay. In vertebrates, including humans, there's no such thing as a true unipolar neuron, which would be a cell body with just an axon sticking off of it. True unipolar neurons are only found in invertebrates, which we don't care about in this class. So we're calling this unipolar neuron, although it's technically a pseudo-unipolar neuron. And I want to mention that because if you're looking at this stuff online and you come across unipolar neuron and pseudo-unipolar neuron, you need to know what the difference really is. Bipolar neurons are pretty straightforward, I think. Okay? cell body has two things sticking off of it. One of them is the axon, and the other one is a dendrite. Okay. Now, we're actually not going to bother ourselves with bipolar cells this semester. There's really only two places in the body that A&P deals with that have bipolar neurons, and they're both in special sensory structures. There are bipolar neurons in the retina, which are in fact called bipolar neurons, that's their formal name, and there are bipolar neurons in the olfactory system, which is your sense of smell. Their dendrites are in the nasal cavity, and their axon goes into the brain to carry information about smells. Okay? So both of those are special senses, which are something that we're going to talk about in AMP2. So other than mentioning that bipolar neurons exist, we're not going to talk about them this semester. And in fact, next semester, we're barely going to talk about them at all. But any examples of them are AMP2 material. And then we have multipolar neurons, which to some extent, it seems like we're saying one thing sticking off, two things sticking off, and then we have a third case. But it's multipolar because the number of things sticking off of it can vary. It could be two dendrites in an axon, which would be three things sticking off of it. Or it could be hundreds of dendrites in an axon sticking off of it. If it's more than one or two things sticking off of it, it falls into the multipolar neuron group. Truthfully, multipolar neurons are what we really are looking at. Pretty much everything's a multipolar neuron except for the couple of bipolar neurons that I said we're actually going to look at this semester, and unipolar neurons, which are always in the same basic structures. Okay, so unipolar neurons are very specific where we find them. Bipolar neurons are in these couple of things that we're not going to talk about this semester, and everything else is a multipolar neuron. How many things coming off the cell varies, and what they look like varies quite a bit, but they're pretty much all multipolar neurons. Now, I want to mention something. It's not in this picture. It is mentioned in the book, but I want to say this explicitly because if you're looking at stuff online talking about these types of cells, you might come across something that's called an anaxonic neuron. And I want to mention this because I think it's a really stupid name. Okay. Now, saying that, I would probably offend quite a number of neuroscientists, but I don't care. What this word means, an is a negative. So anaxonic, whoops, I keep doing that. Anaxonic would mean without an axon. That's impossible. Every neuron has to have an axon. What anaxonic neurons really are multipolar neurons that we can't tell which one is the axon. That's really what it is. There's some evidence that it could be that there's a bunch of processes sticking off of it, and various ones can act as an axon, but only one will act as an axon at any given time. Okay. It has to be that way for the direction of information flow. Okay, so I mentioned this here, and really it's a type of multipolar neuron. It's just you can't tell which thing sticking off of it is the axon. Okay, so it wouldn't be so obvious that there's this one very different thing coming off of it and all of these little other things that look the same. It would be a bunch of these things just all sticking off of the cell. So it's multipolar. There's multiple things coming off of it. It's just it's not obvious from looking at the cell which one's the axon. And there's some evidence that which one's the axon might vary depending on what's going on in the brain at any given time. Okay. I want to mention those because they do get mentioned, and they're actually getting mentioned more and more in describing nervous tissue these days. And so I want you to be aware that it's out there. I'm not including it in here because I don't think it's really a separate type of neuron. The other reason I'm not including it in here is because where that type of neuron would exist is in parts of the brain that we don't really study in this class. Okay, Where they're found is mostly in parts of the brain where high-level cerebral processing, high-level cognitive processing is taking place. And while I'll mention that idea, we're not going to get into how that works at the tissue level at all. So we really don't need to worry about that kind of neuron. And to tell you the truth, I don't really believe it should be considered a separate type of neuron. It's really just a multipolar neuron that looks a little funny. But so those are the types of neurons. I want to go to the tissue now to look at what these types of neurons look like. Okay. And, okay. So it's not in the week 10 folder. In the week 11 folder is a link to nervous tissue. Now, since I'm here and it's in a week folder I wasn't expecting, let me make a point here. The lab quiz that has to do with this material is in the week 11 folder, okay? But this lab quiz is actually about the stuff that I'm covering today and the stuff I intend to cover next week. But like I was saying earlier, I don't think I'm going to get to the lab material next week, okay? So I'll probably have to change the due date on this lab quiz because we won't have finished everything. That the lab quiz is over by the 21st. So if next week we don't get to that material, I'll change the due date to reflect that. But as it is now, it's in next week's folder because I'm ideally planning on getting to that. So I have the nervous tissue link in next week's folder because that's material that has to do with the lab quiz material. But I actually want to talk about it tonight. So if you want to look at this tissue that I'm about to show you, it's in the week 11 folder, not the current week folder, but next week's folder. And it just takes us to one of these box pages where I upload a bunch of these things. Okay. Now, I'm going to start by showing you a cross section of the spinal cord. And actually, let me pull up a picture real quick of what the spinal cord looks like in cross section. This will be good. Okay, so here's a spinal cord that's been cut in cross section. In the tissue, we have what we refer to as white matter and gray matter. Now, this is not actually white and gray matter as we see it because we've stained the tissue. But white matter stains lightly compared to gray matter that stains darkly. In the spinal cord, we have this basic arrangement of the tissue. The white matter's on the outside and the gray matter's on the inside. The shape of the gray matter varies depending on what level of the spinal cord we're looking at, and probably best represents some sort of weird ink blot test. Okay? I'll look at something like this and I'll see, that kind of looks like two bunny rabbits holding hands or something like that. Which might tell you more about my personality than necessarily what we're actually looking at here. But that's the spinal cord. Now, if I go back to this micrograph, we're looking at a spinal cord in cross-section, but we're only looking at a little bit of it, okay? So notice this circle in the middle, and there's a line going up, straight up from that circle in the middle, and a line going straight down. And then we can see this darker staining stuff in that kind of shape, okay? That is basically this much of the spinal cord, okay? So a little circle in the middle with a line going straight up and a line going straight down. And then the darker staining stuff in the middle that kind of has this weird shape off to the side. That's what we're looking at, okay? So the gray matter actually stains visibly darker. We can see the boundary between the gray matter and the white matter very clearly there. Okay? Within the gray matter, we can see these really dark spots, and those are the neurons. Neurons stain really, really darkly. And they stand out very well. But there's more sort of background coloration to the stain pattern too, also, which shows us the full extent of where the gray matter is. Okay. Now, the terms gray matter and white matter actually refer to, and the book describes this in some detail, refer to what unstained tissue looks like. White matter has a lot of fatty tissue in it, or I should say lipid-rich tissue in it. It's not fatty in the sense of adipose tissue, but it has a lot of lipids in it. And so it has a very white appearance. The gray matter isn't actually gray per se. That term is just used to offset the difference between white and this stuff. Gray matter will tend to look kind of pinkish because of the blood in that part of the tissue. Or sometimes I've seen tissue that's maybe a little old and it's aged and it starts to look kind of tan or something like that. But it's the not white part of the tissue. And when we stain the tissue, while it's not gray and white, we can definitely see a very stark difference between the lighter staining and the darker staining parts in there. Okay. And so we're looking in the gray matter. And what's characteristic of gray matter is that that's where the neuron cell bodies are found. And that's what these dark things are all around. So we want to look at one of these in higher, we want to look at into the gray matter in a higher magnification, which is, there we go. Here's the gray matter shown at a 10x magnification. And what we can see here are individual neurons that stain darkly, and then the rest of the gray matter has this kind of light blue kind of staining appearance to it. In the neurons, we see a very clear oval, which is the nucleus. Okay, it's actually lighter than the surrounding tissue because of how the tissue is stained. It's a weird, special kind of stain for nervous tissue, and that's what it looks like. We're used to the nucleus as being the darkest part of the cell, but actually here, it's the lightest part of the cell. Okay. But in the middle of the nucleus, there's a little dot, like we can see right here, called the nucleolus, which really means little nucleus. But what it is a bit more specific than that. Okay. So let me go to the higher power here. So here's one of those cells, and we can see the clear space, which is the nucleus, with the dark spot in the middle of this nucleolus. Okay. And then surrounding it is the rest of the cell body where the cytoplasm stains in this kind of typical splotchy pattern that we see here. Okay. Now, let me just define nucleolus for you, nucleolus, which means little nucleus. What it is, it's a dense region in the DNA where ribosomes are produced. What are ribosomes? Anybody know what a ribosome is? That's a very kind of tangential definition. But I'll come back to why it kind of applies. But anybody want to get closer to what a ribosome is? Ribosomes are the organelles that make proteins. So to say it has something to do with enzymes is fair because enzymes are proteins. Okay. Also, it's fair because a ribosome is kind of like this super enzyme, but it's not made out of protein. It does have some protein in it, but it's primarily made out of RNA, ribonucleic acid, which is why it's called a ribosome. Okay. Now, DNA is the genetic code. We translate DNA into RNA. And where the nucleolus is where the genes for ribosomal RNA are found. So the RNA is translated there, and then it's assembled into the ribosome in the nucleolus. And so we get this nice dark spot there because there's a lot of RNA and it's picking up a lot of stain. Okay.
So the nucleolus is where the genes for RNA is found.
Not just any RNA, specifically the ribosomal RNA. Okay. Ribosomal RNA is different from messenger RNA or transfer RNA. Okay. It's just where that's found specifically. The ribosomes are made right there, but then the ribosomes are shipped out of the nucleus into the cytoplasm. Okay. So really this splotchy staining that we see out here is where there's a high density of ribosomes in the cell. Okay. So that dark spot there, there's a lot of ribosomes. Where it's a little lighter next to it, there's not as many ribosomes, et cetera, et cetera. That's what we're really seeing in this stain for the neuron. Okay. The fact that ribosomes are the primary thing that we're staining here has to do with the way that the stain works, but it also highlights that neurons make a lot of proteins. Okay. Protein production is very important for how neurons work for two reasons. One, a lot of proteins are necessary for the neurons to have their particular shape. Dendrites have a skeleton shape to them, which is, a skeleton scaffolding within them, which is made out of protein. And then there are ion channels and neurotransmitter receptors and enzymes and all these things that are responsible for all of the functions of the cell. There's a lot of ribosomes making a lot of proteins all throughout the cytoplasm. So it stains very darkly there. Okay. So this is what we see of a neuron. When you're looking at these neurons, you're not responsible for saying exactly, are they multipolar, bipolar, or unipolar? Okay. You are responsible for saying they're multipolar, okay, because that's what they are. The next slide I'm going to show you will show you unipolar cells, but everything else we look at today is multipolar cells. Now, when you look at one of these things, like this one that I've been pointing at, you can see something sticking off here, maybe something sticking off here, and maybe something sticking off up here, suggesting at least 3 things sticking off of it. This one over here, where we've cut off center so we don't see the nucleus, but this is another neuron, we can definitely see something sticking off there, something sticking off there, something sticking off there. So there's definitely 3 things, and maybe there's another thing sticking off there. Okay. But if we look at a cell like this, we could say, okay, yeah, there's something sticking off there, and there's probably something sticking off there. But we only see those 2 things. That does not mean that this is a bipolar neuron. This is a multipolar neuron. All of these are multipolar neurons. It just happens to be that we're not cutting through the correct part of the cell to see all of the processes coming off of it. You just have to trust that all of them are multipolar neurons. It's the law, so to speak. Okay. Think of it this way, which is a little bit gruesome. Okay. If you wanted to determine how many limbs somebody had sticking off their body by cutting a 2-dimensional section through the body, you're not going to be able to tell that for any section. You have to take a frontal section through the body, so you see an arm coming off of either side, and a leg coming off of either side. But if you take a sagittal section through the body, especially right on midline, you're not going to see any of the limbs. Okay, so based on a mid-sagittal section, you would say the body has no limbs attached to it. The section through this particular cell makes it look like it only has a couple of processes coming off of it, just because of where we're cutting through it. Okay. So, okay, we'll get the idea of cutting bodies up into little pieces out of our minds and move on. But all of these are multipolar neurons. Now, these really big cells here that I'm pointing at are neurons, but some of these little cells are neurons also. Okay. The size of a neuron really corresponds to where its connections are. These neurons that are in the gray matter of the spinal cord that we're looking at, these big cells, these are the neurons that connect to skeletal muscles. Okay? So when I defined a motor unit last week as one neuron and all of the muscle fibers it connects to, these are those neurons. So this neuron controls one motor unit, this neuron controls one motor unit, this neuron controls one motor unit, this neuron controls one motor unit. How big the cell is corresponds to how many fibers it connects to, muscle fibers it connects to, and how far away they are. Okay, sort of like the unipolar neuron I mentioned before that has an axon that goes from the tip of my big toe all the way up to my brainstem. There's a motor neuron sitting at the base of my spinal cord right here. Again, the lumbar level of the lower back. Its axon goes out and goes all the way down my leg and around my ankle to my toe, and it controls a muscle in my big toe. Okay, so it has a very long axon, and it might connect to, I'm guessing here, 40 or 50 different muscle fibers. Okay. Might not actually; those are kind of small muscles, so they don't have a lot of fibers in them. But so that could be a pretty sizable cell like this. Because it's got to make all of the proteins necessary for the entire cell, all the way down to where it makes its connection. All of those neurons are made here, so it's a really big cell. Okay. But some of these smaller cells here, they might have axons that just go to one of these big neurons. Okay. So they don't have to support a very big cell, and they are small. Also, some of these cells in here aren't neurons, they're glia. And in fact, probably glia outnumber the neurons by a pretty significant factor. Okay. So these are some examples of neurons. Multipolar neurons in the spinal cord. Now, I want to show you unipolar cells. Where'd it go? This is a 4x objective magnification of a ganglion. Now, this particular ganglion is called a dorsal root ganglion, which is what the name is up here. Dorsal means posterior, and then root is referring to the nerve root that's connected to the spinal cord. So the dorsal root is the posterior part of the spinal cord. And then the ganglion is attached to that. The cell bodies that are in this ganglion, their axons are in that nerve root. And the nerve root goes out into the periphery, and the axons go into the spinal cord to synapse at various levels there. Okay. Now, at this 4x objective, we can actually see one really big cell right there. Okay. And sometimes we have really big cells because they're supporting axons that are going that whole distance. They're going from like the tip of the toe all the way up to the spinal, the brainstem. Okay, so they can be so big that we can actually see them under 4x objective like that. But when we move to 10x objective, I don't know if this is that same cell that I was just showing you. I can't remember where I took this particular picture from in that larger picture, but we can see this cell, and there are some other lighter staining cells of similar size in here. They just don't stand out quite as much. And then there are smaller cells. Now, we can't really say that these are necessarily smaller cells. We'd only know that if we know that we're cutting through the middle of every cell. So this little thing right here might be as big as this cell right here. We're just cutting through at a weird angle. Okay. But some of these are definitely smaller cells. And the best way to tell when you're looking at the center of a cell is when you see the nucleus in it. Okay. So we can see the nucleus in this little cell right here, and we can see the nucleus in this sort of medium-sized cell here. We can see the nucleus in this big cell here. So those are probably 3 different size cells. And again, the size relates to the connections. Okay. So we need a really big cell to support the axon that goes 2 meters from the tip of my toe all the way up to my brainstem. But we don't need a very big axon to support, sorry, I said that wrong. We don't need a very big cell body to support the axon that might be coming from the skin right here in the side of my abdomen going up to my brainstem. So that axon there is probably half a meter long or something like that. So it's going to need a smaller, or it won't need as big of a cell. It can get by with a smaller cell like that. Okay. Now, we can see the cells that are kind of clustered together in a row here, another row here, and there's a row there. There's a cluster here and a cluster there and there. In between all of this kind of wavy, fibrous looking area, these are the axons. These axons are coming off of these cells. They're these pseudo-unipolar cells. So there's one thing coming off the cell, and the axon is passing through. One end of it is going all the way out into the periphery. The other end of it is going into the central nervous system. Okay. So I was just showing you these two slides that are labeled dorsal root ganglion, okay, 4x and 10x. The 40x objective slide I labeled unipolar cell because now we're looking directly at a cell with very little of the surrounding tissue visible, okay? But we can see that fibrous looking stuff running next to it. So there are axons going through here. One of them is probably the axon from this cell. We don't see the axon coming off of the cell because the chances of cutting right through that part of the cell is pretty slim. Generally speaking, I don't usually see the thing coming off the unipolar cell. Okay, but unipolar cells are always found in this kind of structure. Okay, it's a ganglion. There are lots of big cells, and there are lots of axons running by. Not all of them are necessarily these ganglia on the posterior side of the spinal cord where the nerve root attaches. They can also be found in other locations, like in the head and neck region. And we'll talk about that stuff in a couple of weeks, but those are unipolar cells. Okay. This is the only example of unipolar cells we're going to look at in these dorsal root ganglia. Everything else we look at is a multipolar neuron. So let me show you another example of a multipolar neuron. And I'm going to show you that in the cerebellum. Okay? Now the cerebellum is part of the brain. It's this little wrinkly part that sits on the back of your brain. And the wrinkles are pieces of tissue that are kind of folded in on themselves. So this is one wrinkle right here. Okay? One little crevice between two folds of tissue. In the cerebellum, there's always this very light staining region on the outside of the fold. This is not white matter, okay? Like we saw white matter in the brain, the spinal cord. This is technically gray matter. It just doesn't stain very darkly. And then we have this very dark, grainy looking part in the center. Okay? These cells are called granular cells, which means grainy looking cells. And then the surrounding part that's lighter doesn't have as many cells in it, so it doesn't stain quite as darkly. Right at the edge between the granular and the lighter region there are a few really big, dark staining cells. Okay? And those are the cells that I want to show you. So this is the 4x objective. This is that at 10x. So here's the granular cells, and here's the lighter staining part with fewer cells in it. And then there are the occasional really big cells at the edge between the two. And here's a couple of those cells with the 40x objective. Okay. Now, these are a special kind of cell that we find in the cerebellum, which is called a purkinje cell. Purkinje is actually a mispronunciation of this guy's name, which is actually like purkinje or something like that. He's, well, he's dead. He lived in, I think it was Hungary or Czechoslovakia, one of those sort of Eastern European countries that are no longer the same country anymore. So like the Czech Republic, Bosnia-Herzegovina, that area there. I can't remember. I think he's Czechoslovakian, but I can't quite remember anymore. But he's a rather famous anatomist. So there are a few things in anatomy that are named after him. Okay? And we just mispronounce his name, purkinje. Okay? This is a large multipolar cell of the cerebellum. And so we're just talking about its anatomy. If you want to remember what it is, that's what it is. That's what it's called. Now, let's see. In the book, so after that sort of cartoon I was showing you before of the different polar definitions of cells, I have this picture here, which I think is actually adapted from a picture that's on Wikipedia or something like that. But these are sketches made from micrographs of different types of cells. And the one in the middle here, this is the purkinje cell. Okay, what we're seeing in the microscope is only the cell body right there, but it has an axon coming off of it, which branches to connect to a couple of different things. And then its dendrites are just all over the place. And really the defining thing about Purkinje cells is this huge dendritic tree sticking out. So going back to the picture here, we just see the cell body there. And that's because what we're using to stain the tissue only stains cell bodies. It doesn't stain dendrites. So we don't see the dendrites, but they're out here. And in fact, going back to the 10x objective version, those dendrites extend all the way out into this lightly stained area. The reason it stains so lightly is that it's mostly dendrites of all of these cells, which doesn't pick up a lot of stain. The little dots that we see in here, some of them are little neurons and some of them are glial cells, but there's not a whole lot. Definitely not as densely packed as the granular cells there. Okay. Now, let me pull up a picture of Purkinje cells because there are some really impressive fluorescent micrographs of them. Okay. So this cell has been dyed with a fluorescent dye. And what happens is it just gets in there and associates with the cell membrane. So we can see the cell body and we can see the dendrites all the way out to their smallest little branches. This is that huge dendritic arbor of the Purkinje cell. And I think that might be part of the axon going off there. And then there are some similar pictures here. This is another one showing a very sort of blocky looking dendritic tree, but it's the same. Here's another one, kind of small. This one has a series of Purkinje cells, all with their dendritic branching trees, showing that they really overlap and fill up all of this space that is that light staining part on the outside. Okay. And then some other ones. This is a computer reconstruction. The colors mean something, probably the diameter of the dendrite or something. And then this is also a computer reconstruction that's been colored so that you can see the main dendrites in the lighter color and all of the smaller dendrites in the bluish kind of color there. So different ways to look at Purkinje cells. These black and white images are, again, ones that are traced from micrographs, kind of like what I showed you in the book here. Okay, this kind of, this kind of thing here. But this has been traced and you see just the outline of the cell, the black and white ones there, the microscopist colored in what you saw there. Okay. But these are Purkinje cells. Another example of a multipolar cell. Now, another type of multipolar cell that I don't have a micrograph to show you is what's represented here. This is what's called a pyramidal cell. It's called pyramidal because its cell body is kind of triangular in shape, and triangles in 3 dimensions are going to be kind of like pyramids. It's one way of thinking about it. So these are called pyramidal cells because in 3 dimensions, the cell body is kind of a pyramid shape. Coming off of the base of the pyramid and the peak of the pyramid are the dendrites. And then the axon kind of comes out of the very center of the bottom of the pyramid. Okay. These are found in the cerebral cortex. Your cerebrum is the big gray wrinkly part that makes up most of the brain. The cortex is the outer covering of that. So these cells are found in there. Okay. We have slides that are supposed to show you these particular cells, but the way that they're stained is not very easy to understand what the cells look like. So I've just stopped using those slides, and instead there's a drawing here in the book, or we could look up, oops, to help us spell it right, pyramidal cell, and there's a bunch of depictions of that. Here's a cell-stained version. This is drawing. These are drawings of cells that have been stained with a very dark stain. Here's one of those dark stain seen in the micrograph. So there's a lot of different variations of what they look like here. But this is a pretty good suggestion of the shape of one of these things. Okay. So I don't have a slide of that to show you in the slide set here because we don't have a good slide in our set to capture. So I'm fine with that. Now the rest of the slides here are of glia. Okay. So I want to switch to talking about glia, and then I'll get to looking at these slides a little bit. So let me go back to the book. Okay. After finishing looking at the neurons, the next thing to look at are glial cells. This table here shows the different types of glial cells by name, okay, and their anatomical division location and their basic function. Okay, so in the central nervous system, there are astrocytes, oligodendrocytes, microglia, and ependymal cells. And in the peripheral nervous system, there are satellite cells and Schwann cells. I've made this table so that similar types of cells are lined up together. Okay, astrocytes in the central nervous system and satellite cells in the peripheral nervous system are both your basic support cells. What they do is different depending on which one we're talking about, where we're looking. But we can summarize what they do as support. For example, what the astrocyte does is it kind of associates a little bit with all the surrounding neurons and helps them by maintaining the solution or concentration of things in solution of the extracellular space. And then very important for astrocytes, they help maintain what's called the blood-brain barrier. One thing that sets the central nervous system apart from the rest of the nervous system is the fact that it has a privileged blood supply. What that means is not just anything circulating in your blood can get into the brain and spinal cord. The astrocytes help regulate what comes from the blood into the brain and spinal cord. You can really only get small soluble substances like ions across the blood-brain barrier, or specifically you can transport amino acids and nutrient molecules across. But other than those, the blood-brain barrier really restricts the movement of everything into the brain. Okay, that helps protect the brain against possible toxic molecules. And a neurotoxin can sometimes just be something that causes neuronal activity and isn't really toxic in the sense of killing other tissues. But overactivity of neurons can actually cause destruction of those neurons. So there has to be this very tight control so that not just anything can get into the brain. And that's based on the blood-brain barrier. Making drugs that affect the central nervous system requires considering the blood-brain barrier. Drugs have to be designed so that they can cross over the blood-brain barrier. So drugs that are amino acids in structure or that can be tied to glucose or lipids or something like that, they can get into the blood-brain barrier. But other drugs that are completely different types of molecules, like antibiotics or protein type drugs like insulin that somebody would take for diabetes, those cannot get across the blood-brain barrier because they potentially would cause problems with the nervous system, and you want to protect the nervous system. Okay, so astrocytes are supportive, and part of that support is they regulate the blood-brain barrier, as well as other aspects of the extracellular space. Satellite cells do kind of similar things. There's not really a blood-brain barrier in the periphery, but there is a little bit of restriction of what can get directly to the neurons because it can cause some problems. But it's not nearly as restrictive as the blood-brain barrier. And it also helps with regulating the extracellular fluid around them. Oligodendrocytes and Schwann cells are the insulating cells. We saw an oligodendrocyte before. That was what was drawn in as the myelin around the neuron we were looking at. Oligodendrocyte specifically means few, which is what oligo means, branching, which is what dendro means, and cell, which is what site means. So it's a cell with a few branches, okay? 2, 3, 4 things sticking off of it, which are going to go wrap around axons and help insulate them. Okay. We find those in the central nervous system. Schwann cells provide myelination in the periphery. One Schwann cell makes up an entire myelin sheath segment. Okay. It doesn't have a cell body that reaches out to multiple cells, multiple axons. The entire cell wraps around one segment of the axon to provide myelination for it. Okay. Now, a very important difference between oligodendrocytes and Schwann cells has to do with tissue repair. Schwann cells will actually encourage regrowth of damaged axons in the periphery, whereas oligodendrocytes will actually inhibit growth of axons in the central nervous system. So going back to what Amanda was asking about the spinal cord injury, part of the reason why spinal cord injury is so devastating is because the tissue itself keeps severed axons from regrowing. Okay? Now, seemingly the reason for that is the connections in your central nervous system are very precise, and you've spent your entire life making them form the connections, and if you change them, it'll cause big problems. Okay? Whereas in the periphery, the connections are a little bit more straightforward. Wherever the nerve goes, it's going to make a connection at the end. So if a nerve gets cut, the axons can grow back and heal the nerve over and remake the connections in the periphery. That does not cause as many problems as what would happen in the central nervous system. Okay. Now, thanks to medical science, we can hopefully direct the regrowth of those axons in the central nervous system. And so the kind of things that can do that would be transplanting Schwann cells in place of oligodendrocytes in the central nervous system, which is one therapy that's being investigated. And then
they would take Schwann cells from somebody else or from?
Probably from the person, kind of like a skin graft, like you take skin off of the butt and put it somewhere else. So it's the same person's tissue.
Possibly make it be able to move again?
Possibly. I don't think that
Maybe
the, yeah, that's probably not going to help anybody that's got problems right now, but it's a direction that the research is heading. Another direction that the research is heading takes advantage of a really weird central nervous system structure that does regrow. The connections between your nasal cavity and your brain that carries the sense of smell, those neurons in the central nervous system do regrow. There's something weird about that they do that. Now, the reason why is because you inhale all sorts of potentially toxic chemicals. And those chemicals could get up through the nerve that carries the sense of smell to your brain. So those neurons that would be exposed to the toxins die off on a regular basis. They have a lifespan of, I don't know, maybe a month or two. And then new ones grow in. And they have to grow through the central nervous system to get to their original connections. Very unusual, but very important for how the sense of smell works. So there's also a line of research that takes pieces of that nerve for the sense of smell and transplants that to other parts of the spinal cord to cause regrowth. I mentioned there is a study that came out sometime in the past year that actually caused regrowth of spinal cord injury. I think it was based on that sense of smell nerve rather than Schwann cells, but I might have that backwards. Again, it's a very early, and I'm pretty sure that was in a rat model or something like that. It's very early research, and the time it'll take for that to be fully worked out and applied to humans is probably decades away. It's decades further along than it was back when Christopher Reeve had his accident, but there's a lot more time to go into that research. But anyways, these cells are the ones that myelinate or insulate axons. Okay, oligodendrocytes in the central nervous system, Schwann cells in the peripheral nervous system. Now, microglia and ependymal cells are found in the central nervous system, and there's no corresponding glial cell in the peripheral nervous system because of the blood-brain barrier. Microglia are essentially white blood cells. Okay? There's some evidence that, in fact, white blood cells during fetal development migrate into the brain and become microglial cells. I haven't seen a definitive answer to that, but there's a suggestion that they are, in fact, white blood cells. Whether they're really white blood cells or not doesn't really matter because their job is the immune function in the central nervous system. So they do in the central nervous system what white blood cells do in the rest of the body. Okay? And it's because white blood cells can't cross the blood-brain barrier in a mature nervous system. So it needs its own set. And then ependymal cells make this stuff called cerebrospinal fluid, or CSF. That's a fluid that's found in the central nervous system instead of basic interstitial fluid that we see in other tissues. Okay? In any other tissue in your body, water is filtered out of the blood into the surrounding tissue, and it makes the extracellular fluid. In the brain, that's not allowed to happen. So instead, ependymal cells filter stuff out of the blood specifically to make this stuff called cerebrospinal fluid, which has less stuff in it than the normal extracellular fluid that you'd find in other tissues. But it's essentially that. Okay. So again, these are only found in the central nervous system because it's an adaptation based on the blood-brain barrier. Okay, we don't need those in the periphery because there's not a blood-brain barrier in the periphery to deal with. Okay, so those are the types of glial cells. Here's a picture of what astrocytes and microglia and oligodendrocytes and ependymal cells look like in this sort of central nervous system kind of tissue here. And then here's a picture of a unipolar cell, which is in a ganglion in the periphery, surrounded by satellite cells and myelinated by Schwann cells. Okay, so this is sort of the peripheral version of this. There's more central glia, so there's more going on in this picture. There's just the two types of cells in the periphery, so we just have them associated with a single neuron here. Okay. It goes on to talk about how myelin forms. So whether it's oligodendrocytes or Schwann cells, basically the cell wraps around the neuron multiple times, making layers and layers of cell membrane, which helps to insulate the cell. In with that cell membrane are some other molecules that make up specifically the myelin. So this is a little bit different than just a cell membrane wrap, but it helps to insulate. This is an electron micrograph of a myelin wrapped axon. Now, electron micrographs are different from light micrographs in that we're seeing a much, much higher power image. This is 1,460,000 times magnification, okay, which sounds a little big to me, but sure. This is from this University of Michigan Medical School website, and so they would have the magnification factor there. It's been a long time since I've worked with electron micrographs, so that seems like such a big number, it's almost hard for me to believe, but maybe it's entirely true. If you look at the myelin, you can kind of see there's a pattern of layers of little black lines. Those are the cell membranes that are wrapped around. But they're wrapped around so tightly that it makes one solid enclosure, okay? And so that's what we're seeing there. And then in the middle, this lighter staining stuff here, this is the inside of the axon itself, okay? I'm not quite sure what this thing off to the side is supposed to be. The label has been cut off, but it's some other sort of cellular structure. I don't think this is nervous tissue here. I think this is a peripheral nerve. But when we're looking at electron micrographs, what we're using to stain is actually heavy metals. So electrons are the, quote, light source. It's not really light, it's electrons. But instead of photons, which are light particles, electrons travel through, which are much smaller, so we can get better resolution. The only thing that'll stop an electron in its pathway is a big, dense nucleus of another cell, big, dense nucleus of another atom, okay? And heavy metal atoms are the biggest and densest stable atoms that we have. So we've attached to the lipids in this tissue a stain made up of osmium, which is a very heavy metal. And then attached to the proteins, which are the little dots inside the axon here or outside here, attached to the protein is lead, which is another heavy metal. We can't tell the difference between lead and osmium because everything's in black and white. But we know in looking at this that this is membrane. So it's lipid. It's associated to osmium. And inside the cytoplasm of the cell are proteins, which would therefore be associated with lead. Okay, that's what we're seeing there. I'm telling you that because we have a couple of light micrographs that I'm going to show you in a second, which are stained with osmium. Okay, so let me move over to that and show you the glial cells. So first off, I want to go back to the spinal cord section. This is just a different part of it. So this is a 10x magnification, but that's hole in the center where there's a line going up and a line which we can't really see here going down, I pointed out before. The hole in the center of the spinal cord is lined with ependymal cells. So these are ependymal cells. We don't see a whole lot of detail to them, but that's what they are. Okay, and that's because that's where we see them. They're not making cerebrospinal fluid here. They're only located in certain places in the brain where they make cerebrospinal fluid. But otherwise, they just act as a lining for the inside spaces of the brain and spinal cord. Okay. They almost look like an epithelium. They're not an epithelium. They're nervous tissue, but they're kind of like an epithelial tissue in that they're a continuous layer of cells lining that space. Those are ependymal cells. I also want to go back to the unipolar neuron slide here. This is the unipolar neuron, really big thing. The nuclei that we see surrounding it, these are nuclei of the satellite cells. And they're actually called that because they kind of look like satellites orbiting the bigger cell in the middle. That's where they get their name. The cell nuclei that we see out where the fibers are, those would be Schwann cells. Not that we can make much sense of them, but that's what would be in that area there. So Some examples of glia there. What I really want to show you is this section nerve here, which I have 2 magnifications, and then this teased nerve here. All of these slides I'm about to show you, 3 slides, are all stained with this osmium. It's technically called osmic acid, but it's an osmium heavy metal stain. Now, before with the electron micrograph, we were using it because it blocked electrons. It blocks photons in the light microscope too. Okay, so any dark black spot we see here is where there's a high density of lipids. Okay. The nerve is actually just this thing up in the corner that kind of looks like a slice of watermelon. The rest of what we have here is a different tissue, okay? Not nervous tissue. What tissue has a lot of big clumps of lipids in it? Tissues we studied before. Adipose tissue, fat, right. When we looked at adipose tissue under the microscope before, it looked like this kind of wispy, spiderwebby looking stuff because we were staining everything but the lipids in it. Here, this is sort of a reverse image of what we saw before. We're staining the lipids and we don't see the rest of the cells there. But this is all adipose tissue. So there's some fat surrounding this nerve, probably helping to protect the nerve a little bit. But if we get into looking at the nerve directly, we see staining this, the myelin surrounding axons. So for some of these, you can see a pretty obvious circle with a blank space in the middle, okay? This is only staining the lipids, so we're only staining the myelin. We're not staining anything else in the cells. So we can see a lot of myelin-surrounded axons in here, okay? I don't think I have a 4DX objective of this. No, apparently I don't for some reason. But at 10x, you can kind of see enough detail to see little round things that are just like what we see at the very high power in the electron micrograph here. So one of these thick outer coverings is one of these things that we see here with a little white space in the middle. Okay. Now, this nerve has been cut along its length, split in half like a banana split, and then sectioned crosswise. So we're looking at half cross sections of this. Okay. Cutting along the center of it, splitting it in half like a banana, is technically called medullating a nerve, cutting through its middle. I mention that because we can kind of see what it is here, and the other slide I want to show you here, this teased nerve was also cut open like that. But instead of sectioning it in cross-section, the individual fibers were teased apart with forceps or pins or something like that, so that we can see an individual axon. But they've been stained with the osmium, so they're nice and dark fibers that we can see. Now, you can see a little spot right there, a little gap. Okay, that's one of those nodes of Ranvier. Okay, we can see another one right there. We can see one right there. We can see one right there. That might be one there. Might be one there. These ones that I've pointed out, that one here, that one here, that one here, and that one here, those are nodes of Ranvier. They look very distinctive. This gap right here is not a node of Ranvier. That's a little tear in the fiber. Okay. So when they look really messy like that, they're definitely not nodes. When they have that kind of sort of definitive end on either side of the gap, that's a node of Ranvier. Over here, it probably isn't a node because it looks like it's sort of a little pulled apart section there. Or down here, I'm not positive that's not a cut in the nerve, but at least one side of it looks like what we see here. Okay. But this particular region I took a picture of, I took this picture because in this axon that we can follow all the way down, we see a node of Ranvier there, and the next node of Ranvier is there. Okay, that's how far apart the gaps are. Okay, so back to the picture of the neuron that we were looking at before. Here, the little myelin sheath here looks really short, but in actuality, the distance from one node to the next is really quite long, relatively speaking, for the width of the axon. Okay. If this were to be drawn to scale, then this myelin sheath would have to be really long and go off the side of the page. Okay. So for the sake of making a picture that fits on the page and shows all the details that we want, we have to make these artificially short. Just keep in mind that really the distance from one to the next is a lot longer. Okay. And the function of those nodes are going to be important when we talk about how the electrical activity works, which we'll deal with next week. Okay. I wanted to be sure to show you those, and I did that at the expense of showing you the spinal cord white matter. Okay. This is a 40x objective picture of the white matter. This is stained with a cell stain rather than stained with osmium to see the myelin. So we stain the axon in the middle and then some of the surrounding extracellular space. And these white circles are where the myelin would be surrounding an axon. So this is sort of a negative image of this. Okay, very different magnifications. But just so I don't have to move off of this picture for too long, the myelin doesn't stain with the cell stain that we're using, just the cellular components of the axon and the surrounding extracellular tissue. Okay. So this is sort of the opposite reverse image of that watermelon slide I was showing you earlier. Okay. So the white matter looks white because all of this clear space here is lipid-rich myelin, and it's going to just appear white rather than pink because of blood passing through, or gray, or whatever color the gray matter is. But that's what myelin would look like in a cell stain. Okay. Just as a counterpart to this slide here. So if this were a negative image, a reverse image, it would basically be what I was just showing you there on the white matter. Okay. So I actually showed you that kind of quickly too. But that sums up showing you the different types of glia. Okay. And that's the end of this section of the book, chapter. Then we're going to go on to looking at the function of nervous tissue and the action potential and how we communicate between neurons. So that's the material that we're going to cover next week. Okay. Like I said, I plan on doing that in the first half next week, and then the second half, we'll start looking at the structure of the brain, which would complete the material for the neuroanatomy lab quiz. It's entirely possible I won't get to talking about the brain next week, and so I'll push the due date for the neuroanatomy lab quiz back a week to give us time to cover that in class. And I'll remind you of that next week. But there's no lab quiz specifically for this week that's due next week. There's nothing in the week 10 folder like that. You can start working on the chapter assignment stuff, but they're not actually due for a couple of weeks because we're going to keep talking about that chapter next week. So, otherwise, have a nice night.